Alia University, Kolkata, West Bengal, India. I am glad to present here our research work at ICIBM conference. The paper titled as A Linear Regression and Deep Learning Approach for Detecting Reliable Genetic Alteration in Cancer using DNA Methylation and Gene Expression Data. First, I will discuss what is DNA methylation. This is a promising biomarker in cancer detection and cancer classification. Normally, DNA methylation is used for single work like single differentially methylated genes genes finding or differentially methylated region DMR finding. But our challenge is to incorporate different factors to the DNA methylation so that we can use it at multifunctionalities for different directions such as prediction of gene expression using DNA methylation, differential expression analysis, cancer classification, half gene binding, and among all others. For this work, we use biomethyl package to do such type of incorporation work. First, till now there is some existing method which we use for the DNA methylation incorporating in the gene expression. But these methods have some shortcomings. Those shortcomings are, first of all, to determine the directionality of the evaluated gene expression estimated from the DNA methylation is difficult. Then it is also difficult for variance in the size of canonical promoters as well as presence of the distal argument which initiates biases into the association of methylated region with gene models. The third point is there is high probability for selecting a long gene in place of overlapping or nested gene. There is some tools also available for methylation data into genomic region formats like GREAT, Galaxy, but use of these tools are also create complication. To overcome this problem, we use the biomethyl package where we use the linear. First of all, we take the methylated data and TCGC cancer data, but we use the biomethyl package where there is a linear regression, and we use methylated data as independent input and gene expression as dependent input, and after that, to extend the biomethyl package, we use empirical based test lemma, and also after that we use the disease classification using neural network and deep learning. First of all, this is our workflow, and in our workflow we use the cervical cancer data. So here our disease is the cervical cancer. What is the cervical cancer? A type of cancer which starts in the cervix. Normally we know this is the omen cancer, a hollow cylinder that connects the lower part of the uterus to a omen vagina. Most of cervical cancers grow in the cells of the outer surface of the cervix. Normally, woman can't understand the initial stage that she is a patient of cervical cancer because the symptoms are very normal in her menstruation time problem. This is nothing but abnormal bleeding during menstruation time or after having sex, pain in the pelvis, as well as pain during the urination. So, in our workflow, we use the cervical cancer methylation data. In the data set, there is a 27,578 genes, and our sample size is 215. And this sample, there are two types of sample. One is uterine cervical cancer samples. The sample size is 63 and normal sample size is 152. After data collection, we use the pre-processing step. In this pre-processing, we use first we eliminate the CPG sites that had missing value in more than half of the samples, And after that we use a traditional quality control R package in mix, which use for discard the outliers and replace the meshing values using the popular k nearest neighbors KNN algorithms. After pre-processing, we use the biomethyl package where we have taken the observed gene expression, TCGA cancer gene expression, and this biomethyl data to integrate using linear regression, and we got the predicted gene expression data. After that, to extend the biomethyl package, we use a normalization, Boom normalization, and we get the normalized predicted gene expression data. Then we applied here the empirical base test using lemma, where we have taken the FDR cutoff, which is FDR lesser than 0.001, and after that we find out, we detected the differentially expressed gene. The number of gene we got 6282. Then we use the classification. Here we use classification tool, which is the neural network based deep learning tool. Actually, we prefer here to use deep learning because deep learning is useful for large data set where the sample size is greater than 1000 also. But here our sample size is 215. This is the medium size data set, but we use this deep learning to make our tools more generalized. So we prefer neural network based learning, neural network based classifier in place of other classifier like random forest, PAM, etc. So here we use NNet. In NNet, first we use 10-fold cross validation on all samples to find out the training set and test set, and then we use deep learning analysis. In deep learning analysis, our epoch value is 100, and after deep learning, after first deep learning analysis, we have computed intermediate performance metrics like true positive, true negative, false positive, false negative, and we have repeated the entire procedure 30 times. So our total procedure actually repeats 100 into 10 into 30 times. So there is no biasness in our classification result, and we we compute the different classification metrics like average accuracy, average sensitivity, average precision, average specificity, average overall error rate, and AUC. Then also we do the another things, which is half gene finding. On our differentially expressed gene DEGs, we applied the Pearson correlation analysis, where we taken the correlation value greater equal 0.8 or lesser equal minus 0.8, and we had selected those edges. We have selected edges among those genes, and then we use the cytoscape for network analysis, and here we use degree centrality measure, and we got top 5 in degree half genes and top 5 out degree half genes. Besides this, we do the gene set enrichment analysis for CAGE pathway and geoterm BP, geoterm CC, and geoterm MF analysis for using web-based gene set analysis toolkit, WebGStat. And we find out all these pathways and geoterm by filtered FDR value lesser equal 0.05. This is our total workflow, and this is our workflow of our proposed work. Then this is the internal steps we performed for this work, and this is our result, differentially expressed gene. I already told 6287 DEGs we got, and this is the top 10 DEGs. I filtered it, listed here, and filtered it by FDR value. My our FDR cutoff is FDR lesser equal 0.001. And this is our classification matrices. Average accuracy 90.69%, standard deviation error 1.97% we got, and also the area under curve is here 0.858, And this is our ROC plots for all classification matrices. Here we also did the comparative study. We use another deep learning tool, which is RSNN tool. And here also we use input cross validation and repeating the entire process 30 times, and here we got the 87.27% accuracy. So we can say that our framework has better performance than all other method using deep learning tool. This is our half-gene network structure for whole net. And this is our top 5 half genes according to in degree centrality measure. This is our top 5 half gene according to out degree centrality measure. And this is our FDR sorted cage pathways, top cage pathways where the FDR value lesser than 0.05. And this is our GeoMF term. So in our work, we provided a framework using linear regression, differential expression, deep learning to provide correct biological interface for integrating DNA methylation and corresponding TCGA gene expression data to uterine cervical cancer. Here we eliminate the outliers and applied linear regression to determine predicted gene expression. After linear regression, we got 6,287 differentially expressed gene through empirical basis using lemma, and we use NNet as deep learning tool. Actually deep learning we can apply for any type of data, not necessarily so the data set should be structured. We can apply it in deep learning in unstructured data set, any type of data set, like we can apply it on audio data, on video data, on image data, any kind of data set we can use deep learning. So here we prefer use the deep learning data, and in it we use the tenfold cross validation, and we also perform comparative study and after we also perform comparative study, and you can say that our opt-in DEGs are strong and efficient in disease classification. We perform in degree, out degree gene network analysis using cytoscape. Also we do the pathway analysis, and our framework is useful for better biological interpretation of the DNA methylation data rather than single differential methylation analysis or differentially methylated region founding. In future, we want to extend our current work through integrating random forest ensemble method into the deep learning strategy to obtain a better classification model in all perspectives, then apply that a big data or single cell RNA sequential data or other TCGA tissue specific data for cancer classifications. This is our references. Thanks for listening me. Thank you. We use linear regression using biomethyl package, and there we have taken methylated data as independent variable, and the gene expression is the dependent variable, and total work is done by the biomethyl package.